Hello again, everybody, and welcome back to Science 7. Today, we're going to look into transcription. So what we know so far is we know the structure of DNA. We know the structure of DNA consists of base pairs, A binding to T and G binding to C. We also know the structure of RNA. Similar to DNA, however, A binds to uracil, while G still binds to C, right? We know that DNA is a double helical structure, while RNA is not. And we also know, we talked about last class, how DNA is replicated, right? How DNA forms another DNA strand that's 50% of its parent DNA strand, right? What we will learn today is we will learn how DNA forms RNA, right? Because if we remember our central dogma, we have DNA forming RNA, which will later go on to form proteins. However, we're just going to look at this step here, the DNA to RNA step, and it is called transcription. Transcription. So transcription is the process, like I said, that converts DNA into RNA. More specifically, transcription makes mRNA from DNA in 3 distinctive steps. The first step being initiation. So transcription begins with the enzyme RNA polymerase. RNA polymerase. And now if we remember from DNA replication begins with DNA polymerase 3. So in transcription, it is RNA polymerase. So RNA polymerase binds to a segment of DNA at a promoter region. At a promoter region. Promoter regions are high in A and T base pairs. Now, why would you think that they are high in A and T pairs? Well, if we remember, what binds together A and T versus G and C? Well, they're both bound through hydrogen bonds. However, how many hydrogen bonds? A and T has 2 hydrogen bonds. Well, G and C has 3 hydrogen bonds. Now, RNA polymerase is going to bind to our DNA, and it's going to separate the strands. So it's much easier for it to separate 2 hydrogen bonds rather than 3 hydrogen bonds. So from step 2, so once RNA polymerase is bound to the DNA, it's going to start forming base pairs. It's going to start forming base pairs in the 5' to 3' direction. So the 5' to 3' direction. Now, if we remember, our DNA runs anti-parallel. The strands run anti-parallel to each other. So if RNA polymerase is forming RNA in the 5' to 3' direction, which strand is it going to be attached to? Well, it's going to be attached to the 3' to 5' strand. And this strand is called the template strand. Conversely, the other strand of DNA is 5' to 3', and it's called the coding strand. So our RNA, our newly formed RNA, is identical to the coding strand because they run the same direction, 5' to 3'. However, our RNA, what differs from RNA and DNA? Well, our RNA has uracil instead of thymine. Uracil instead of thymine. Other than that, the strands are identical. And as RNA polymerase moves through the DNA, the stuff that it already coded for, the DNA back here, is going to recoil. It's going to recoil. And how does that differ from when we when we replicated DNA. Because remember, when we replicated DNA, there was something stopping the recoiling of the previously synthesized DNA, right? SSBs, remember those? They are not present here. So since they are not present, we have the recoiling of DNA. And finally, step 3 is our termination. So once we have a sufficient RNA strand, it's going to have to leave. It's going to have to detach from the DNA. And in doing so, it's also going to have to release our RNA polymerase, Both of these, once detached, can go on to bind to other DNA strands and to continue translation. Great, so now what? Now that we have our RNA strand, now what? Well, transcription's done, and now we're down here. So this is our mRNA, our messenger RNA strand. Our messenger RNA strand goes through translation to get to our proteins, right? And our proteins are just amino acid sequences. That's all a protein is. So how does mRNA get translated into proteins? Well, proteins are three base pairs in length. So every 3 base pairs, they code for a protein, for an amino acid. And these 3 base pairs, they're called codons. So let's say A, U, G. So each 3 base pairs codes for a amino acid. But if we remember, there's only 20 amino acids, right? It's only 20 amino acids. How many combinations can we make of 3 base pairs given the fact that we have 4 bases? Well, it's simply 4 times 4 times 4, right? Each position could have one of the 4 base pairs, or the bases. So we could have 64 different combinations of codons. However, if codons code for amino acids and we only have 20 amino acids, what's happening there? Well, the base pairs, when they form codons, they could form also what's known as start codons, and stop codons. And as the name suggests, this is where translation is going to begin at the start codon, and where it's going to terminate is at the stop codon. However, that doesn't account for all 64. So the 64 codons could make more than, or could be coded for more than one amino acid. So we could have, let's say, 3 or 4 different codons for an amino acid. So that's just a general overview of the transcription and translation process. There is more biochemistry that goes into it. However, this is a good introduction, and this is where we'll stop for today.